you can hear me, I'm going to go ahead and get started. Sam, if you can try presenter mode right now, perfect. So I'm going to introduce myself, introduce the logistics for this meeting, and then introduce Sam. First of all, my name is Mike Smansky. I'm an associate professor in the biochemistry department, and I'm going to be the moderator for today's talk, which is by one of my students. So it's a unique privilege for me to introduce my own student today. Thank you all for attending. Thank you for being robust enough to figure out this Zoom, which is a new mechanism for us with the MAISRC showcases. We're presenting this as a webinar and it is being recorded. The attendees in today's webinar don't have the ability to be seen or heard. And so we would like you to ask your questions using the chat function, which if you scroll with your mouse to the bottom of the screen, there's a little text bubble with the word chat beneath it. There is another bubble with a Q&A, but we would prefer that you make questions and comments in the chat function. That'll make it a little bit easier for us moderators. We have one place to look to for questions. I will be recording all the questions. Feel free to upload questions to the chat at any time during the talk. I will collect those and I will ask those questions to the presenter at the end of his presentation. So with that, I will go ahead and introduce our speaker today. Sam Erickson got a bachelor's degree in chemistry from Macalester University in St. Paul. And for the past 3 years has been a master's student in my group at the University of Minnesota, where he's worked on this project to engineer a new and frankly paradigm shifting approach to controlling invasive species, namely carp. Sam has been a great member of the lab. He's a bit of a polymath. He's an expert in too many different fields for me to keep track of. And you'll see that come to bear in his presentation today. He's had to master fish husbandry, genetic engineering, DNA construction, and among others. Currently, Sam is a PhD student in the biochemistry, molecular biology, and biophysics program. And the subject of his talk today is something that we call engineered genetic incompatibility for the biocontrol of carp and other invasive species via genetic engineering. Sam, with that, I'm going to turn off my video and microphone, and the floor is yours.
Very well. Good afternoon. Thank you for sharing your time. Thank you for sharing your attention with us today. I'm Sam, and I'm developing male birth control for fish. Our freshwater ecosystems here in the Midwest are facing a severe threat from the invasion of carp. And engineered genetic incompatibility, or EGI, it's a new technology in development at the University of Minnesota for the control of invasive fish species. For the purposes of this talk today, I'm going to make an assumption that may or may not be true for you, but I'm going to assume that you're at least aware that there is a carp invasion and that you believe we ought to do something about it. If you want to have a bigger, higher level discussion about the ecology of carp and say the economic consequences of an invasion, there are many other experts on this very subject, and probably maybe in this room and elsewhere at this showcase who can talk about those subjects. But for today's the precious time, I want to focus our attention on the cutting edge science and engineering that makes this particular technology possible. I anticipate in the room we probably have a few government folks, maybe some fish and wildlife professionals, and while this EGI technology will need a few more years in the lab to mature, I anticipate that down the road, we may be having conversations in the real world about how to implement this technology, and those conversations will be a community effort. As such, it's important to me that you know a bit about this technology. I think there's a virtue in having some shared vocabulary and a grasp of some of the technical fundamentals. So that being said, let's get into how this idea works. In this figure, I'm trying to make a special fish. I'm trying to engineer a fish, and in this figure, I've shown it in purple. And this fish will not be purple in real life. This is just for illustration purposes. The engineered fish will actually be completely indistinguishable from a wild fish, with one key difference, that when our engineered fish attempts to reproduce the wild fish, it results in embryos that don't succeed in developing to make mature adults. The vision is that using an integrated strategy of a combination of existing physical methods with a controlled wild release of our engineered EGI fish could result in either complete control or eradication of carp with a method that's low cost and has no direct impact on the surrounding ecosystem. We're aiming to develop this system with common carp right now. We anticipate, however, that'll work just as well in any invasive species of carp in North America. Before we move on, I want to highlight two technical advantages of this particular strategy of applying a technology of this sort. One of those is that notice that our engineered EGI fish are really incapable of having any offspring. They're not going to succeed in reproduction in the wild. And as such, the long-term environment, the environmental impact of the wild release of an engineered fish of this type is limited to the lifespan of that fish that you release. The second point that I want to point out is that one advantage of this approach is that notice that if you have two engineered fish, if you have a female and a male engineered fish, those can actually succeed in reproducing. As such, to make this happen, we start by retrieving a wild fish and then doing our genetic engineering workflow to produce our engineered fish. But that step's quite technically complicated to do, but we only need to do it once. If we do this once successfully, then the children of these fish can actually be propagated indefinitely in any normal commercial aquaculture setup. So it makes it a particularly scalable approach in that the cost of making EGI fish is essentially the main cost is a one-time cost of this initial development, initial production step. In terms of the mechanism, the key concept to understand here is that EGI disrupts gene expression in a developing embryo. So in this approach, we're not releasing any toxin. We're not, the approach is that we're disrupting gene expression. So let's talk about how a fish grows. Every fish starts its life as a single cell, which then splits and splits and splits and eventually produces this mature larvae with billions of cells. Every cell in the body of a fish has exactly the same DNA. Has the same set of 52,610 genes in carp. And the way that we see how cells differentiate, how we have a spine and a brain and a heart and eyeballs, it's based on differences in which genes are expressed, that which genes are activated and used, that in every cell only a subset of all those genes are used. And the successful development of a fish requires very precise control of all 52,610 genes. Among those genes, there are a few key regulatory genes whose job is to direct the expression of other genes. They're sort of the conductors that control the overall flow of information. In our system, we're trying to sort of hijack and exploit the precision and the delicacy of this approach here, that we're trying to disrupt the expression of key regulatory genes. I want us to look for a moment at what a gene is. A gene is a length of DNA that has some information in it that altogether works together to encode all the information needed to make one thing in a cell, one molecule in a cell. And over the past 100 years, we geneticists, we've gotten really good at reading DNA and interpreting it. And one thing we've learned is that DNA has a grammar, it has a syntax. When we look at a gene, there's actual distinct sections that have different purposes in the same way that you have different words in a sentence. It's like learning a language. On we're looking at a double-stranded length of DNA here. And on the right with this arrow, we call this this is what most people think of as the gene popularly. It says the coding DNA sequence has the information to make one molecule. But there's something pretty cool about that upstream, immediately before the coding DNA sequence, there's another sequence that goes along with it called the promoter sequence. And the information in this sequence isn't actually manifested in the final product, but instead this information encodes when and how much of this molecule to make. How often are we doing this? What tissues do we need this? This is the sequence that's not the only factor, but it's a major factor in controlling which genes are being expressed in which tissues at which times. So our approach looks at the gene, and we're going to focus specifically on using and exploiting the fact that our genes of interest, our crucial regulatory genes that we want to disrupt, have a specific promoter sequence that we can target and not target the rest of the genome. Well, before I move on, I should make one more point here. The way we're going to disrupt the genome is by overexpressing a gene. If we pick a key regulatory gene that needs to be maintained at a very specific expression level, and then we're going to, maybe it needs to be at a 1 or a 2, we're just going to turn it up to an 11 and disrupt the embryonic development. This approach has actually been shown to work with great success by my colleagues in our lab in yeast and insects. First, I want to show that our first success was with yeast. My colleague Maciek Maselko demonstrated this. He did this, well, let's just look at it. So on the left here, let's see if I can play this. This is a video, it's supposed to go. And right now it does not seem like it wants to cooperate. So let's just move on from that.
So Sam, I'll interject. If you turn off the red laser pointer, it'll give you control to start a video. And we can still see your cursor, so it won't really impact your ability to point things on the slide.
I see. Let me see if I can make that happen. Ah, there we go. Good. Thank you for that. Good suggestion there. So on the left, we're seeing normal, healthy yeast budding happening. This is what it's supposed to look like. Yeast are a wonderful model organism because they're a microbe, but they can also engage in sexual reproduction. So you can do very quick, useful genetic experiments that way. And on the right here, this is our engineered yeast strain that's been engineered to overexpress a gene called actin. It's a crucial, structural gene. And instantly we see the cells don't quite look right. There's something wrong. The cells started to produce so much of the structural protein, it actually just exploded. So this was the first demonstration, proof of concept. And since then, my colleagues have been having great success developing this concept in insects. And so much so that they're actually beginning started a startup company to try to commercialize this concept. As for my work, I have 2 projects running in parallel. I'm trying to develop this concept in common carp, and at the same time, I'm using a model organism, zebrafish. Common carp are quite difficult to work with genetically. They're not a commonly studied organism in the genetics world. On the other hand, zebrafish been worked with for a long time, and we have a lot of colleagues at the University of Minnesota who are quite skilled in doing genetics with them. So this would be analogous to, say, if you wanted to study cancer in humans, you might want to start with a mouse, right? Start with a mouse model organism, and then move on to studying your disease in humans. It's the same idea. I'm starting with a small, easy to work with fish, and then moving up to a large fish on the very plausible assumption that the results that we get from zebrafish can be translated into carp. To build EGI in zebrafish, I need to essentially make 3 modifications. I need to introduce what I'm going to call 3 genetic components, 3 parts. And I'm going to turn on my laser pointer again. And so the first one is this molecule. We call this our programmable transcription activator. It's a molecule that we can program to target a specific gene in the genome of the developing zebrafish embryo and overexpress that gene, perturb its expression. There's a second part, the single guide RNA, which is a piece of RNA with a particular sequence that allows us to completely specifically target our PTA molecule to a particular gene in the genome. If you have parts 1 and 2 together, the system activates and it'll find its gene of interest and drive lethal overexpression. So what we want to do is we actually want to prevent that from happening prematurely. So we add a third component, which is this resistance mutation. We pick that crucial gene for developmental regulation in our fish in the laboratory. We use CRISPR to induce a gene edit at our target site to prevent our system from activating, essentially a safety on our system. Then something special happens when you mate a wild type in an engineered fish. You actually bring together 3 components that are not present in our engineered fish, namely the guide RNA, the PTA, and a wild unedited target gene. And the combination of these 3 will have this reaction that will prevent the embryos from developing. At this point, we've actually succeeded in characterizing all of the key genetic components for EGI in zebrafish, all 3 of these parts. And the vision is the point here is that part of the joy of genetic engineering is that we're not actually making these molecules. We're not putting these molecules into fish directly. We're actually putting in pieces of DNA into fish that encode these molecules. So it's the fish that's doing all the work. It's making these molecules. And by making these sections, these loops of DNA that have our particular genes, we can make actually quite precise edits and changes into our fish by adjusting this transgene section that we're introducing. Ultimately, the first 2 parts are going to be introduced with a double transgene cassette. It's a piece of DNA that we're inserting. And then this third part, the resistance mutation, is actually an edit to an existing gene. So sort of like 2 steps, introduce 3 pieces. At this point, we've selected candidate target genes. We found a PTA molecule that expresses well in zebrafish. We found a good way to express our PTA. Based on editing this transgene cassette, I can actually have some control over where, when, and how much these molecules are being expressed in the fish. And we found a particular system that seems to work really well for this approach by using an endogenous gene, this DMRT2A zebrafish gene, as our expression system. So I'm not going to go into all of this in depth about these previous steps. I want to share just a few highlights from some of our more recent work in characterizing the single guide RNA that will allow us to target our molecules to a particular gene. Before I move on from that, I want to leave you all with one sort of a major concept here. And the concept is about where genetic engineering components come from. That I'm talking about these molecules here. For instance, this is our programmable transcription activator molecule. And what I want you all to understand is that the parts here they weren't invented. They weren't built from scratch. They were all things found in nature that biochemists and geneticists saw and imagined some sort of application for. So there's a precedent for this, a very long precedent for this in human history, of humans going out into nature and finding things from other forms of life that are useful and could be repurposed. So I like to imagine maybe a long time ago, one of our ancestors might have been walking out in the steppes of Eurasia and saw one of these animals and thought, wow, that's an interesting animal. That's a really strong, fast animal. I wonder if I can ride that. I wonder if I can do something with that. And now, many thousands of years later, now we have a long heritage of breeding horses with strong legs and strong backs for carrying people and tools and whatnot. And similarly with fur, that at some point, some human a long time ago saw a big furry animal and thought, I'm cold and that animal isn't, and maybe there's something interesting I can do with that. Even though a horse was not evolved to carry a human, and a caribou was not evolved to make humans warm, some clever humans realized we can repurpose that. And in the genetic engineering community, we're doing the same thing. We're just doing it with molecules, and that's a little trickier to explain sometimes. So, for instance, our PTA engineered molecule here is actually composed of several subunits, several sections that have been fused together. And these subunits come from different aspects of different, kingdoms of life and whatnot, and they have different purposes in their original organisms. But clever biochemists look at these and realize that you can do something cool with them. So, for instance, one of the key components that makes this whole system possible is this blue blob here, which I'm showing here, a more detailed, not a cartoon version, but a more detailed three-dimensional chemical structure model. This is the component that allows our system to bind specifically to a particular sequence of DNA that we've chosen. That molecule was first identified, first discovered actually by scientists at Danisco. These were yogurt engineers who were trying to understand why some batches of yogurt had bacteria in them that were getting sick and other ones were not. And those questions ultimately led to the discovery of this molecule, which is actually part of the innate immune system of a bacterium. So if any of you had yogurt today, you might have this molecule floating around in your stomach right now. So these are existing natural known molecules that have just been now attached to other molecules in a clever way to allow you to do something you previously couldn't do. Similarly, even our system for getting the gene for this molecule into our fish is also a discovered molecule. It's Tol2 transposase, which came from this Japanese rice fish, and it's as part of this natural system for moving pieces of DNA and sort of recombining its own DNA. And some geneticists saw this system and realized that this could be used for fish genetics, that you could trick the system by disguising your DNA between two sequences that this molecule recognizes and tricking this molecule, and then to introducing your gene, your transgene, into that organism system for recombining its own DNA. So moving on, more recently, the hurdle to overcome was improving the precision of targeting the system. The way we do that is by screening for single guide RNA. And there was a hurdle to overcome that with the current state of the art in the technology, designing single guide RNAs is a little tricky. That normally what you have to do is you have to make several of them and test to see which one works. It's very hard to predict exactly which one's going to work the first time. The physical chemistry of the binding is quite complicated. Ideally, what I would like to do is I'd like to replicate the success that my colleagues had in the insect team, in which what you actually do is you generate a large group of various transgenic adult lines of zebrafish with these various components and cross them together and see what happens. There's a barrier to doing that in that because of the 2-month regeneration, 3-month regeneration period, that doing an experiment like this, testing it, planning it and setting up and testing it is actually a 9-month process. So to overcome this, I came up with this shortcut solution to do sort of a quick and dirty experiment, in which you can get results in 1 week, where you inject an embryo with a transgene cassette that has multiple parts in it and the same piece of DNA, and then watch that embryo develop to maturity and take notes on that. The advantage is that you get quite rapid results. There's a number of disadvantages that ultimately you don't have precise control over the exact level of expression of that transgene. The way I overcame that, though, was well, we'll get to that there. The point of this here is, though, is that this system did seem to work, that we were seeing lethal overexpression of the gene. In this case, I'm targeting a gene called GATA5. It's a key regulatory gene that patterns it's one of the first genes that steps in when the early embryonic heart is forming, the heart tissue. And here we're seeing images of embryos about 6 hours post-fertilization. Here's one injected with the transgene cassette. Here's one with the control. And right around this period of 8 to 24 hours post-fertilization, we see that this is what a healthy embryo is supposed to look like here, that a very large number of these embryos will start to crash out, that they fail to get beyond this key sort of barrier right around the 8 hours post-fertilization step. There's something intriguing about this particular time. This was a very encouraging result of the timing of embryonic death, is that it corresponds to this window in the stage of embryonic development. What we've done here is we've cross-listed existing data from another group that was measuring the expression of various genes over time, genes of interest for our system. And in this case, what we're doing is we're using this gene expression promoter to drive the expression of the programmable transcription activator system. So our system, based on these results here, we expect that our system should be activating and ready to go right around the 8-hour, right around this spot here, right when expression of this gene is starting, right at the beginning of somatogenesis. And that's in this window here from about 8 hours to 24 hours, and that's the exact window when we're seeing embryonic death. So I was quite encouraged by these results, and motivated by this, decided to scale up this experiment a lot. And there's a lot to see on this figure. I don't have time to go in depth about everything. The key is that we have this, we'd like to test various single guide RNAs. So we insert this transgene cassette, this is a piece of DNA, we inject it into embryos, and then we watch for a week to see what happens. So we targeted three target genes, targeted 1, 2, 3, 4. For each gene, there was 4 candidate single guide RNA designs. And what we're seeing here is a time series showing the rate of healthy development over time. So we want these lines here to be going down. That's indicating that our fish are not succeeding in reaching adulthood. In this case, we did find a few single guide RNAs that perform substantially better than others. And all of this data at the top has been summarized below with weighted averages for each target gene, the 3 target genes, and our single guide RNAs. So over on the right here, we have our controls. So for instance, if you inject, if you take a plate of about 50 embryos and you don't touch them, you don't inject anything, approximately over 80% of those embryos are going to reach maturity. On the other hand, if you inject one of these 2 single guide RNAs targeting the gene GATA5, you get less than 10% development. Considering that with this approach, we're actually probably getting a less than 100% dose, maybe a 0.5%, maybe a 50%, maybe 25% dosage of this transgene cassette, of this expression from our transgene cassette. That's actually very encouraging initial results. The next step is what we like to do is like to build the whole system. Since we've characterized the 3 parts separately, we like to bring them together. And so we've planned a 5-generation, 10-month breeding scheme summarized here that involves both crossing steps and 2 microinjection steps. One to do a CRISPR mutagenesis to mutate the endogenous gene, and one to introduce our own transgene. And as such, what we expect is probably by this time next year that we'll actually have some, we'll actually have a working zebrafish EGI strain to test. So that brings us up to where we are now. We have sort of 3 main ongoing projects. The one I mentioned is we're going to be building this complete zebrafish EGI model using parts that have already been separately characterized. We're just pulling them together. The analogy I make is it's as if we're building an airplane, and we've done due diligence, and we've tested a propeller, we've tested a body, we've tested a wings, and now we're just going to have to put them all together and take it for a test spin. Next step is what we'd like to do is we'd like to take these results and move these into carp. So that's been going on in parallel, but these results are a good starting point for further research. For instance, we'd like to use these results to inform genetic design considerations. There's one very interesting challenge in doing this, and that goes beyond the scope of this talk. But overall, zebrafish is a very good genetic model for carp, except for one big detail. That big detail is that the common ancestor between zebrafish and common carp is hypothesized to have lived about 400 million years ago and split at some point. And then during the course of evolution, we resulted in a zebrafish genome on one hand, but about 8 million years ago, that's very recent in biological time, the ancestor of the common carp underwent a whole genome duplication. So something kind of went wrong, something interesting happened, and those kids had twice the number of twice the number of chromosomes that their parents had, and that gave them some sort of perhaps some sort of advantage, and as such, all the common carp that we have in the world now have twice the number of DNA. And this poses some, from an engineering perspective, this is a frustrating, annoying thing. From a scientific perspective, it's a wonderful opportunity to learn some interesting fish genetics. So that's an ongoing question. And then additionally, down the road from here, we have a one-of-a-kind carp farm. I developed this system called the ABC system. It's a novel system for doing carp husbandry specifically for collecting eggs and sperm out of season, so that we can do, we can do genetic experimentation with embryos, not just once a year in spring when they want to reproduce, but rather we can induce our fish to reproduce out of cycle. Additionally, we're developing better systems for mutagenesis, transgenesis, for micro-injecting eggs, for getting DNA into carp cells. That remains an ongoing challenge. For all three of these projects, a lot of these are going to be led in the next several months by Nick Jacobs, who has joined our lab as a full-time technician. And I think that's probably enough for today. I'd like to thank in particular MAISRC for funding this work, and for all the staff at MAISRC who make everything easier. My master's degree is hosted by the University of Minnesota Biotech Institute. I was co-advised by Dr. Smansky and Dr. Beyer, and then also want to give special thanks to Nick Jacobs, who's been joining and taking increasing leadership for some aspects of this project. And at this point, I think I want to open up for questions. Thank you very much for sharing your afternoon with us.
Thank you, Sam, for that talk. As Sam mentioned, we'll open it up for questions now. And I wanted to remind you, or tell some of you who joined late for the first time, the way to ask a question is to click the button in the center bottom of your screen that says chat. And that will bring up another window that will allow you to type a question and send it to all of the panelists. So some of you have your hand raised right now. I'm not sure if that means you have a question to ask, but if you don't mind, please submitting your questions via the chat box feature. That will be the best way for us to field those and forward them to Sam. So Sam, while we're waiting for more questions to come in, I thought that I could get things started by asking the first question. Your approach is to learn how to build the system in zebrafish before engineering the carp. Can you speak a little bit more to the similarities and differences between zebrafish and carp that will impact your ability to port this system directly from one type of fish to the other, and maybe
Certainly.
from extension then from carp to other types of invasive fish?
Certainly. Let's see if I'm going to go back. How do I do that? There we go. So that's here in sort of this second ongoing project area. So one, there's a lot of tools we can build off of. There's a lot of existing knowledge to be used that can make that happen. So zebrafish in common, one advantage is that, a few advantages here. One is that it was about 4 years ago, the entire common carp genome was sequenced by a team at a fisheries organization in China. I sequenced it, and there's a lot of existing bioinformatic tools, computational tools that we can actually use to compare genes between zebrafish and common carp. As such, almost essentially every gene, most of the genes in zebrafish have what's called an ortholog in carp. They have a gene that has a shared common ancestry. And because of the speed, the very slow speed at which DNA sequences change over time, we can actually with great precision identify those relationships between the orthologs. One advantage of this particular system and how to do that in carp is that our target genes are these key crucial regulatory genes. So for instance, one of the best candidate genes is this gene called GATA5, and it's a regulatory gene that binds to DNA and directs that DNA in the expression of other genes so that the early heart tissue can form. And that gene is an extremely conserved gene. Every vertebrate, every vertebrate has this gene, and such zebrafish has it, and as such, carp has it as well. And as such, if we believe that disturbing the expression of that gene in zebrafish will work, it seems very plausible that disturbing the same exact gene in carp will work. There's a caveat, though, as I mentioned, in that common carp 8 million years ago, their ancestor had this whole genome duplication event. So they actually have two GATA5 orthologs. The word that we use in the genetics literatures, they're called onologs. So we have these two possible candidate target genes. And at this point, our plan is to test both of them because we know that they're the onologs because of the bioinformatics method. But trying to pick which one's going to work better, it's very hard for us to predict exactly which one just because these genes haven't been studied in depth in carp. So the ultimate goal is we think it's very plausible that for every good target gene that we find in zebrafish, there will be we can find in an onolog that has the same corresponding functionality. So that's sort of the foundation of our logic to do that. Additionally, a lot of the existing DNA pieces, the plasmids, the pieces of the transgenes that we have been shown, the components that we're using in zebrafish weren't first developed in zebrafish. They've been shown to work in every vertebrate. For instance, our PTA molecule works it works in almost every kingdom of life, not quite, but as such, we're optimistic that the components and our target genes will ultimately work together. And we're doing our best to leverage every detail that we gain from our zebrafish study to inform further carp experiments.
Thank you, Sam. Another question from Matt in the audience. What is the regulatory process for releasing a genetically engineered fish into the wild? And a follow-up question to that is speak towards the timeline of doing this. Okay. And I'll say if you need to phone a friend for any of the details on regulatory processes, you're welcome to do so.
Very well. So there's two questions. The first one is, what is the regulatory and what is the timeline? And ultimately, those two questions are very linked because I anticipate that the regulatory process, that'll be a rate limiting step in making that happen. Where should we start with that? So I'll start with a recent success story, a certain precedent, a place to start with in terms of regulation. That several months ago, the FDA announced their first approval of a genetically engineered animal for human consumption, for eating, and it's a fish. There's a company called AquaBounty that has a genetically engineered Atlantic salmon they've produced. The reason you want to engineer an Atlantic salmon is for aquaculture. You want to make larger fish that ultimately, if you make a bigger fish, that your energy input cost per fish in terms of your energy input per pound of meat that you're selling, fish meat, goes down and whatnot. It just becomes a more efficient process. And that's an ongoing thing. So that's a good starting point. It took them many years to overcome that regulatory hurdle there. So in terms of working with the FDA and with genetically engineered fish, there's a platform to start on. How much further beyond that, I'm not sure. Issue then again is we're not just farming these fish. The idea is to put them out into the wild, and that goes beyond the realm of the FDA, that goes elsewhere. And that very quickly gets beyond the limits of my knowledge there. So I anticipate regulatory issues are going to be substantial and for good reason, that there's a reason that we have these organizations. And I'm sure we'll work with them to make sure that we have a technology that works completely and is very safe. And it'll be part of my job to characterize these technologies in great detail so that can be happened. The second point is about timeline. So in terms of timeline of regulation, that goes beyond my expertise. But in terms of timeline of developing the system, it's my very, very realistic hope that by this time next year that we'll have a working EGI model fish to demonstrate perhaps at this showcase. Additionally, there's going to be some intermediate steps in the next 10 months for testing and refining the system as we go. So I'm optimistic about making this system work in zebrafish. In terms of making it work in carp at this point with current methods, that's several years away, maybe many years. There's an additional side project that I didn't mention on a slide here because for time limits that we're looking to dramatically accelerate that process. But with our current workflow in zebrafish, that's going to be many years. And for that, the reason for that is the rate limiting step is that to bring the components together, you can't just do it in one step. You actually have to produce fish separately with these components and then bring those together. And that's due to the technicalities of how you breed the transgenes together. So in terms of carp, I don't see that in the near future. It's going to take several years of breeding to make that happen. In terms of more details of regulation, I think there's probably other people in this room and maybe else at this showcase who are probably can give a more detailed account of how that might work.
Okay, thanks, Sam. And yeah, I'll say to Matt, if you want to follow up more on the regulatory path or process, I'm happy to follow up with you offline.
Mm-hmm.
Another
Mike
question, Sam.
To
have you engaged the general public, and how comfortable are they with this approach?
That's an ongoing project there. So not this year, but last year, we've done a few things. We started this year. One of our better approaches has been last year we actually had a booth at the Minnesota State Fair, and we're doing talking with people who are visiting our booth and taking surveys to get a general sense of everyone's feelings about this. And so we have actually a data set with these results. And I'm a scientist by nature, and I'd rather give you data sets than general trends and whatnot. So maybe I think in the future we'll help publish these results. One general trend that we have seen that I don't have the data on me right now to demonstrate is that generally when we meet people outside the lab, the more that they're aware of the carp issue, the more that they know about invasive carp, the more that they're willing to consider the virtues of this particular approach. If you believe that we need to do something about the carp invasion, that carp pose a substantial threat to our lakes, to our rivers, to our freshwater ecosystems, and our sort of Minnesotan way of life, then you look at the options available. And one thing you'll notice is that with existing methods, none of them are, they're not so effective sometimes. And the ones that are effective are either really expensive or they involve hurting surrounding environment, releasing toxins or whatnot. This approach promises to be relatively low cost, and actually substantially lower cost than other approaches. And we think it's going to work very well. And because of the way that's genetically encoded, it's completely species specific. So there's no direct immediate impact on the surrounding environment. But involving the community long term is it's a priority for us. So I've spoken in many other sort of non-scientific settings. I talk about this project both with the genetics world, but then also presented this at the Midwest Fish and Wildlife Conference, spoken at local government, sort of what do they call them, sort of our local Minnesota freshwater regulatory agencies, spoken at even the Rotary Club and whatnot, to begin to start that dialogue. What we'd like to see is some more awareness of techniques like these and to have a informed, honest discussion. I think that the real world, work of actually making this happen is several years away, but it's coming up pretty quickly. So it's time for us to prepare to have that interdisciplinary big picture discussion for sure.
Great. Well, thank you again, Sam, and thank you to the audience for participating today. It is 12:46, and we want to respect your time and give you a chance to transition to the next talk you're going to observe. So I wish you all well, enjoy the rest of the showcase, and have a great